We're going to talk about protein structure and function, primary structure of proteins, secondary structure, tertiary, quaternary, and then protein folding. I love protein folding. I love all of it actually, but so proteins are biologically active. Proteins are polymers of amino acids, and just as you were learning to draw, they are linked by peptide bonds, which are amides. And there are many conformational, so you get these long chains, and proteins, they can be hundreds of amino acids up to 1,000 amino acids long. So you have these really long chains, but when they're functional, they're globular. So they have to fold into a very specific structure. And they're just beautiful. The precision of the fold and how it happens so quickly is really impressive. And when you have a protein folded in its proper state so that it can function, we call that its native conformation. That's the active form. And it's generally accepted that that is the lowest energy conformation. And so they have these compact structures, but also at the same time, a lot of proteins have large segments of random structure, like loops that are flexible and move. That are somewhat undetermined. So some things are locked like into a helix or a sheet, but then there are things that move and sample different, what we call conformational space. And there's different levels because they are pretty complex, right? So you are used to looking at molecules that might be about 100 grams per mole, right? Proteins are 200 to 300,000 grams per mole. So they're much, much bigger, more complicated. And so there are different levels of looking at the structure. The primary structure is simply the sequence of amino acids. It is the order that the amino acids are linked together in their sequence. And we often call that the covalent structure because it's determined by the covalent bond formed in the peptide bond. And we always read it amino terminus to C-terminal end, carboxy terminus. The next level of structure is secondary structure. And this is, there's a couple different flavors of it. It's the ordered three-dimensional arrangement of the backbone. The backbone of a protein is everything but the R groups. The R groups we consider we call them the R groups. If we're referring to the backbone, it's that NC alpha part of it that I was talking about. So the backbone is everything but the R groups. And the secondary structure is mostly formed with hydrogen bonding between peptide bonds. And there's really only a couple kinds or flavors that you have to know. There's the alpha helix, and there's the beta sheet. And beta sheet can be parallel or anti-parallel. There's a few other minor parts, but for our purposes, alpha helix and beta sheet, parallel and anti-parallel, are the secondary structure we look at. And then the tertiary structure is the three-dimensional arrangement of all atoms in the protein. So we look at how elements of the secondary structure fold back on itself. So maybe there's a helix and another helix, and the identifying the helix would be talking about the secondary structure, showing how the helices come together and stack. That's tertiary structure, and that is usually stabilized by the R groups on the amino acids. And now quaternary structure, that is when you have separate polypeptide chains that come together to form dimers and trimers. So we say that they associate. So if you say that something has a dimer or a trimer or a tetramer, that is referring to the quaternary structure. Okay, primary structure, it's the amino acid sequence. That's really all there is to it. And what we see a lot in the news these days is, right, the word for 2021, variant, right? Variant is a different sequence in the spike protein on the coronavirus. So that is a change in the primary structure of the spike protein. Before variants became part of sort of the popular press, we would usually refer to them as mutants. When there's a substitution in an amino acid in the sequence, that is a mutation or a mutant. I don't know, maybe the press decided that was too scary, and so they started calling them variants. Okay, so what's really, really amazing is that when you have simple proteins, for a lot of proteins, all you need is the three-dimensional structure to determine the fold of the protein. So Chris Amundsen famously did this experiment where he worked with ribonuclease H, and he made it he made it chemically. Instead of having a cell make it or isolating it from a natural source, he used peptide synthetic methods and linked the amino acids from N to C terminus. So you have this long chain of amino acids that we know has to have a very specific three-dimensional fold in order for it to work. And what he found is that he made it in a tube, and it functioned just like the native ribonuclease H. So that means all the cellular components where the ribonuclease H is made, that protein, are not necessary to determine the three-dimensional fold. And the only thing that's different in amino acids are the R groups. So what that tells us is that the R groups on the amino acids are enough to determine the fold of the protein. It's not always true, but it's true a lot of the time. And when it's not true, we can usually help it a little bit to help it find its native state. So that was an incredible experiment that proved a lot, that if you, and that's why we can clone things, because when we make a protein in its non-native location, it still very often folds into the right form. It's just a physics problem. But the other thing that's interesting, there's so many possible forms. We know that it's like, if you were to sample space and calculate all the possible conformations of that ribonuclease, it would take years for it to randomly find the right fold. So we know that there's also a pathway. It doesn't randomly sample conformational space. There's an actual folding pathway. Okay. And so I actually don't like that this is in here. Most mutations are silent. There's lots of mutations that happen in various parts of proteins, and they have no effect on the protein. But if you hit one in an important spot for folding or for function, sometimes the changes in one amino acid sequence can alter the biological function. And the example that the book goes over is hemoglobin, with that amino acid substitution that changes the shape of the red blood cell and causes sickle cell anemia. But the vast majority of changes or mutations in the amino acid sequence are silent. It really matters where it is. And actually, what's interesting, I've been following these, it's variants, following their substitutions. There are definitely hot spots on the spike protein that matter and other spots that don't. But when they say something is the delta variant, there are often they're just looking at a few key positions. They're not looking over the entire sequence. So there are likely to be other mutations but they're just classifying it as a Delta variant because it has the classic that are associated with the Delta. Oops. Okay, now secondary structure is mediated by the backbone, right? So that means the peptide, the CO and H of the peptide bonds mostly. And it's generally, it's the hydrogen bonding arrangement of the polypeptide backbone. Each amino acid has 2 bonds of free rotation. And these slides are kind of like, it's easier to show. So this is talking about the Ramachandran angles. Okay, so this is showing, get oriented, here's an alpha carbon. And so on either side, there is an NH and a CO. So this is our peptide bond here. The green box is the peptide plane. This is the alpha carbon of the neighboring amino acid. And here's the peptide plane on the other side. So here's another peptide bond. So on either side of every alpha carbon in a protein, there are 2 peptide planes. If you go back and look at the peptides you drew, you will see peptide bonds on either side of the middle amino acids. And the peptide bonds, remember, they have that partial double bond character, so there's no rotation there. But you can rotate between C alpha and the carbonyl carbon, and you can rotate between the C alpha and the amino group of the other peptide bond. And those rotations have very specific names. They are phi, that's what the symbol is, and psi. And they are dihedral angles because it's the angle that plane moves. Ramachandran was the first guy he would create a plot of, I forget which is X and which is Y, but a plot of phi versus psi. And when they all come out in one spot, it's the alpha helix. And then, of course, I'm going to get this all wrong because it's been a while since I've looked at, when they come out in another spot, it's the beta sheet. And so if you know the phi and psi dihedral angles for for each amino acid in the peptide backbone, you can fold the protein. You know what the fold looks like. So my very first protein society meeting at the Manchester Grand Hyatt, I saw Ramachandran come out of a talk, and I got super excited. It's like, Ramachandran, that's Ramachandran. Well, then somebody told me, and I had an Indian student laughing at me. I remember the first time I told this story in my chem lecture at USD. Ramachandran is like Smith in India. So there's like tons of Ramachandrans. It wasn't the Ramachandran. But I did actually go to a party in Point Loma with Chris Anfinson, the guy who did the ribonuclease experiment, and he got a Nobel Prize for that. So that was kind of neat. And that was really Chris Anfinson. So anyway, those are the phi and psi dihedral angles. And just to put it in something that might look a little more familiar, and the way I remember is C psi, if it goes, it's this angle here, and if it goes towards the carbon, it's psi, C psi, and the other one is phi. We don't spend too much time on that. I spent a ton of time on it in my research, but okay. So now we're looking again more at secondary structure. One of the options is the alpha helix, right? And so in the alpha helix, what you are looking at here, oops, actually it's a little bit better on the next, I'll show you. The alpha helix is stabilized by hydrogen bonds. If you can see here, this is in structures, nitrogen is blue, oxygen is red. So this is the NH of one peptide bond, and this is the CO of another one. And they hydrogen bond to each other, and they hydrogen bond in exactly, it's like I to I plus 4. So if you're in a helix, amino acid 1 will hydrogen bond to 5, 2 to 6, 3 to 7. There's a very specific pattern that you can look that's characteristic of the alpha helix. The other thing about the alpha helix is that it's got a right-handed helix. Let me go to the next slide. So this is just showing the same alpha helix on its side a little bit bigger. There's the hydrogen bonds. There's one there. And that's what stabilizes it and also makes it so specific. Because if it unwinds just a little bit, those hydrogen bonds won't line up. So it's always exactly 3.6 amino acids. Remember, residue is another term for amino acids. So it's 3.6 amino acids per turn. Now, this is one of the things I miss about not teaching in person, because I used to have fun with this in class, is I used to hand out telephone cords to illustrate helicity. I have some pet peeves, you know, as we get older, we get like a little bit inflexible. And one of my pet peeves is when I see a left-handed helix. Like, first of all, they just bother me because they don't occur very much in nature. They're almost always right-handed. But when I see it in a scientific setting and it's meant to show something that should have a right-handed helix and it's left-handed because they've like flipped the slide, it really makes me mad. So if you look at this helix and you point your thumb, let's use your right hand and you point your thumb down the barrel of the helix, your fingers will curve, and they'll curve in the direction of that ribbon. Try it with your left hand. So if you try with your left hand, my thumb goes up the helix and my fingers go to the back, but that ribbon right here is in the front. So this is a right-handed helix, and that only works with your right hand, right? So I point my right thumb down the axis of the helix, my fingers curve towards the back, and the ribbon goes towards the back. The other way I am able to tell this is when you have a right-handed helix, you look at the parts that are in front, it's always higher on the right-handed side. Like if I were to rotate this up, actually go back this way. If you, right here, the parts that are in front are here and it's higher on the right-handed side, that's our right-handed helix. If it were left-handed, it would go like this instead of like that. I went to symphony at the Salk, right? And I love going there. And that's where Francis Crick retired, right? He did the structure of the DNA alpha helix, which is right-handed. And so the symphony is performing and they have two slides on either side projecting of this on the screen, and they have little helices. And I was so mad because one was right-handed and one was left-handed. I was like, if Francis Crick were here, he'd be so angry right now. Okay, so alpha helix, each peptide bond is trans. By trans, they mean the O is on the opposite side of the NH. This is trans. A cis peptide bond would look like this, and they almost never happen. I don't know of cis peptide bonds in proteins. Okay, and then the other thing that's worth pointing out, so let's say I have my helix, all the hydrogen bonds are they're in the tube of the helix. Let's say now I turn my helix and I'm looking down the barrel of it like that. The R groups point out from the sides. So they are not involved in the secondary structure. The secondary structure is determined by those hydrogen bonds. But now that the R groups point outwards from the helix, they are available to stick to other helices or maybe other beta sheets. So the tertiary structure comes from interactions sticking out from the tube of the helix. More pretty pictures. Side chain sticking out, that's a better representation. Okay, there are things that can disrupt an alpha helix. A proline, because it's got that ring in the backbone and it doesn't have the NH. Remember we went over, it doesn't have that H on the peptide bond. It's not available for hydrogen bonding. So prolines disrupt alpha helices. It's pretty rare to find a proline in a helix. And if you do see one, then the helix often has a little bit of a bend in it. If you have really large amino acids, they're bulky. So like a tryptophan next to a leucine, those would bump into each other and also destabilize the helix. If there are electrostatic repulsions, so having two positive charges near each other, like lysine and arginine, or two negatively charged amino acids near each other, like glutamic acid and aspartic acid, those repulsions, negative, negative, or positive, positive, could destabilize a helix. And then the steric. Sterics refers to the bulky nonpolars. Okay. And so here's an example where we have, this looks like an aspartic acid. This one looks like a glue, and we have negative charges next to each other. And so those are going to repel. Oops, repel. And so they would destabilize the helix. Okay, the other major flavor of secondary structure is the beta pleated sheet. And in this case, we have sort of crinkled sheets. And the peptide chains are adjacent to one another. So here's two examples of beta pleated sheets. This is in the N terminus is here. If you look at an alpha carbon and the amino group is to the right, that means the N group is that way, the amino terminus. So this is running in this direction. And then again, here's an alpha carbon, and then the N is to the right. So this chain is running in that direction, N to C. Because they are running both from right to left, that is parallel. And parallel parallel beta sheets result in these diagonal hydrogen bonds. Down here, it's telling you, but if you look at the alpha carbon, the N is on this side. So this one runs in this direction and this one runs in that direction. Because they are head to tail, that is anti-parallel. These are considered to be more stable and they're more common than parallel. And they think it's because the hydrogen bonding lines up closer and more linear. So it's got better hydrogen bonding patterns. It's more common.
So how do we know the direction of the arrows? Do we just have to figure out like the colors, like what is what?
Yeah, so here's a peptide bond, right? What's not in the peptide bond is your alpha. So if you're looking at the backbone and you have an alpha and the N is on this side, that means the amino terminus is on this side. If you look in the peptide backbone and you see the N to the right of the alpha carbon, it's going to run in that direction.
Right. When I look at these structures, I just like don't see it.
I think the biggest handle is the N for nitrogen is blue. This is a different representation of the pleated sheet. Anti-parallel. Okay. Now there are two major classes. Well, there's lots of different kinds of proteins, but the sort of big classes are fibrous ones, which have a structural role, and then globular ones, which have usually a different kind of function. Enzymes are globular. Other things can be globular. But fibrous ones have structural roles, meaning like they actually, like keratin is a fibrous protein and its structural role is to form hair. It's also in rhinus horn and your fingernails. It forms like scaffolding. And fibrous proteins are characterized by having repeating primary structure. So remember the amino acid sequence is the primary structure. And what happens with fibrous proteins is they have a repeating pattern that gives rise to this structure. And they usually have one type of secondary structure. They're usually long fibers or large sheets. Silk fibroin. Silk is made up of silk fibroin, and that is mostly beta sheet. Wool is made of keratin. Oh yeah, so structural roles, keratin in hair and wool. And this is mostly, this is helical. It's a coiled coil. And because it's a helix, it's kind of springy, right? You can imagine a helix behaving like a spring. So wool is more springy than silk. Because if you look at a beta sheet, it's already pretty extended. You can't really stretch it much longer, right? Then it just goes flat. But if you look at a helix, you can stretch it further before it becomes linear. So fibrous proteins that are characterized with helices tend to be more springy and more stretchy than ones with beta sheet. So they are mechanically strong and they are insoluble in water, right? Hair does not dissolve in water. Wouldn't that be horrible? Collagen is a fibrous protein. So, and it is a triple helix. It's so water insoluble fibers. They consist of 3 polypeptide chains that wind around each other, and the repeating amino acid sequence that you get is X-pro-gly. So X can be any amino acid followed by proline followed by glycine, or X-hydroxyproline glycine. So there's a 3 amino acid sequence that repeats a lot. So every third position is glycine. And this is a cartoon of what the structure looks like. So there's 3 different polypeptide chains and they twist around each other. So it's a coiled coil and they call it the collagen triple helix. And so with age, these collagen helices become more and more cross-linked. There's cross-links between the hydroxyproline and hydroxylysine. And when that happens, there's increased cross-linking. Your collagen gets less elastic. It becomes more stiff. And then you get wrinkles. So until you can Okay, so that's fibrous proteins. So they are in massive abundance, right? Because there's lots of collagen and there's lots of keratin, but there's not that many different kinds of them. There's a lot more different kinds of globular proteins, but they tend to be in less quantities. So globular proteins, these are ones that are spherical. It's one where the protein backbone folds. Instead of being a long fiber, you get a ball where things fold on the inside. There's a defined inside and a defined outside. And in general, so think of a sphere, a solid sphere. On the outside of that solid sphere, you're going to have the polar amino acids. So which ones can you think of that could be polar? I want a new color. Be brave, you can type it in. A polar amino acid. Anything with a charge. Yep, aspartic acid. Oh, that's ugly. What did I do with that? Aspartic acid, glue, his. Yep. Also the serine, because it's got that OH group. And doesn't have to be charged, so glutamine, right? and asparagine, those would also be polar. There's other examples. Threonine is pretty polar. Okay, then also so polar on the outside, and that's because it's in contact with water. Otherwise, yep, tyrosine would count too. Single letter Y. So polar on the outside, that's so that it has favorable interactions with water and dissolves. And then the inside is hydrophobic, it's non-polar. So that would include, what do we have for non-polar amino acids? Yep, valine. Leucine, that's where I was headed, yep. Phenylalanine. Isoleucine.
And as a refresher, the nonpolar amino acids are the ones with like primarily the methyl groups as side chains. Is that the right way?
Yes, yeah, there's a lot of methyls, but not all of them have methyls, like tryptophan does, but it's CH.
Got it. Okay.
Yeah. Okay, and so the globular proteins, so remember, like, keratin is a coiled coil of helices. Collagen is a triple helix. That's pretty much the only secondary structure. Silk is mostly beta sheet. Most globular proteins will have both helix and sheet. Not all of them, but they generally have mixtures of alpha helix and beta sheet. And they tend to be soluble in water and in salt solutions, and they have compact structures. So this is just showing a comparison of something that's fibrous and something that is globular. And this is showing myoglobin. I think every biochemistry textbook uses the same exact spherical proteins to start with for their structures, and it's always myoglobin. I think it's because the first structure solved was sperm whale myoglobin. Okay, so now tertiary structure. So we have helices and beta sheets. If you look at this myoglobin here, it's all helix, there's no beta sheet. But see how you have helix here, here, here? That's the secondary structure. But how they come together, like this helix orienting near that one, that is described by the tertiary structure, like how the elements of secondary structure come together. And it's generally mediated by the side chains, the R groups of the amino acids. And the tertiary structure is the three-dimensional arrangement of all atoms in a molecule. With secondary structure, you can describe the orientation lavigal. Oh, I like that. Are those all of the, well, we've got two L's though. Maybe lavid, yeah. Secondary structure, you can describe the arrangement of the backbone. If you have the tertiary structure, you can describe the arrangement of all the atoms in a molecule. So it includes the conformations of the side chains. This is kind of repetitive, I think. And so how these things come together are through non-covalent interactions. That's why we go over that in that first homework. So think about things like dispersion forces, hydrogen bonding, dipole-dipole interactions, generally between the side chains. So hydrogen bonding, hydrophobic interactions. You could have electrostatics. So you could have a lysine potentially interacting with a glutamic acid. That would be an ion pair. And then often metal ions are incorporated, and that stabilizes the structure. And then disulfide bonds between cysteines. This is the only one that's covalent. Disulfide bonds are covalent bonds. Everything else is non-covalent. And the ones that would be in the interior are going to be where the hydrophobic amino acids are. So it'd be hydrophobic interactions are generally going to be on the interior. Yeah. So backbone would be on the surface, hydrogen bonding with polar side chains, because the polar amino acids will be on the surface. Hydrophobic will be interior. Electrostatic, you will find on the surface. When you do see something that is charged and buried, it's usually really important. It's there for a reason because there's an energetic cost to having it there. And then disulfide bonds. So this is just to give you an idea of how much a disulfide bond can help. Phospholipase A2, this is the enzyme that I studied as a graduate student. It's pretty small, 119 amino acids is a small protein, but it's got 7 disulfide bonds, which is a huge number. And those 7 disulfide bonds are all represented with these yellow lines. They make this protein stable to 100 degrees Celsius. Think about putting an egg in boiling water, right? The egg white becomes solid. That's the protein unfolding, the lactalbumin in the egg white. That's what causes it to go solid. But phospholipase A2 would actually be just fine because it's got its 7 disulfide bonds. This came from, the one I studied came from the Indian cobra venom. Its job was to poke holes in cell membranes so the toxins could get in to its victims. Oh, and it's also worth pointing out is that disulfide bridges, so the cysteines, when you see them just by themselves, are the free sulfhydryl form. When they are in a disulfide bond, there are 2 cysteines that come together. This is reduced, this form. When you have a disulfide, it's in the oxidized form. Oops, bridge. Now, it turns out that the interior of the cell is a reducing environment. So if you have an intracellular protein, it will only have this form of cysteine. Oxidizing environments are extracellular. And I think in the lysosomes and the Golgi. So there's specific organelles where you can have disulfide bonds and extracellular. Okay, this is a cartoon illustrating the different kinds of structures in proteins and the kinds of stabilizing interactions. So here we have the helical structure with the hydrogen bonds, beta sheet, again with hydrogen bonds. You can have a metal being complexed. So often what you'll see is if you have something like a calcium, the calcium is positively charged and you'll see carbonyl oxygens pointed towards it to stabilize it. And zinc, it's often NHs, I think, would be, and not NH, but Ns. This is showing hydrophobic interactions between nonpolar amino acids. This is our disulfide bridge. And then this is side chain hydrogen bonding. So those are all kinds of interactions that could work. Okay, got a couple more minutes. So X-ray crystallography is one method for determining protein structure. So basically what you have to do is purify the protein, get it super, super pure, because only pure things make crystals. And then you have to find conditions where the protein will form crystals. And that in itself is like a field. People specialize in being able to grow protein crystals, and they are worth a lot of money, like that, having that capability, because everything starts with knowing what the structure looks like. And so, and in order to get the structure, you have to have a crystal. And so they'll usually have like 96 well plates where they're systematically vary conditions to see which ones will give them crystals. And then you take your crystal and you put it on a sample holder, and you bombard it with X-rays, and the X-rays scatter according to the positions of the atoms in the protein. It's actually 1 over it's called inverse space, because if an atom is at XYZ, the X-ray will scatter at 1 over X, 1 over Y, 1 over Z. And from the patterns in the scattering, they can deduce the structure of the protein. So this is a diffraction photograph of glutathione synthetase, an enzyme. And from this, they can get the 3-dimensional arrangement of the atoms in the protein. My advancement to candidacy exam was it was a paper that presented using anomalous scattering to solve the phase problem in X-ray crystallography. It was absolutely brutal, and only the person who assigned me the paper understood it. Which is a bummer, because I had to answer questions for like an hour and a half, and he was satisfied in 10, but they had to come up with other stuff, which eventually got me. Because I was, like not at all comfortable speaking in front of groups, let alone 4 faculty members. So I froze. I got through it, but it was not fun. And then the other method for, oops, I want to go back. The other method for determining, sorry, you can't see the top, structure of proteins is NMR spectroscopy. That was my specialty. That it's not as efficient as X-ray crystallography, but we're better able to change conditions. Like, we don't have to have the crystal, so we can do things like put in an inhibitor and see what it looks like, and change the temperature and see how the motions move, and all the kinds of stuff like that. So we take things that look like that in 3 dimensions and assign all those little peaks to each hydrogen and carbon and nitrogen in the protein, and then solve its structure. This is alpha lactalbumin. So here's an example of a globular protein. It has helices. There's a little one, and then it has 1 beta sheet. Is this parallel or anti-parallel, this beta sheet?
Anti-parallel?
Yeah, it's anti-parallel, because it's going in this direction here and that direction there. Anytime you see a sheet that's separated by a turn, it has to be anti-parallel. The only way to get parallel is to like have it be like, here's your ribbon, right? Then the protein does something else, and then it comes back that way. That would give you parallel. Okay, I think I will stop there. We're gonna look at myoglobin and fetal hemoglobin next time. And it's a really interesting story. I would recommend reading, and then definitely watch that one video that was in the lesson plan, because that's going to help you understand a lot more about the fetal hemoglobin. And I will just say,